Welcome to The Explainer. Today, we are breaking down the pitcher method for breast milk storage. Now, if you're a lactation professional or you're studying for your IBCLC exam, this is for sure a topic you're going to want to know inside and out.
So let's get right to the big question. Is it actually safe and effective to pool all your breast milk from different pumping sessions over a whole 24 hours? We're going to dig into the science, check out the official guidelines, and look at how it works in the real world to get you a clear answer. Let's do it.
First things first, let's make sure we're all on the same page. What is the pitcher method? Exactly. Let's start with a really simple definition. Okay, so in a nutshell, instead of dealing with a whole bunch of tiny little bottles cluttering up your fridge, you're just pulling all the milk you pump over a 24-hour period into one single container. The whole idea is to make life simpler. The process itself is super straightforward. You pump your milk like you always do. Then, and this step is optional, you can chill that fresh milk in the fridge for a bit. After that, you just add it to your main pitcher. And at the end of the 24 hours, you prep the bottles you need for the next day and pop the rest in the freezer. Easy, right? Okay, so that's the how, but what about the why? What does the science actually say happens to the milk when you mix it all up like that? This is where we get into the really critical data on nutritional consistency and, of course, safety. And wow, this chart right here just illustrates it perfectly. This is from a key 2013 study by Stellerwagen and their team. See all those little squares? They represent individual pumping sessions, and calorically, they're all over the map. But look at the dark ovals. That's the 24-hour pooled milk. See how tightly they cluster together? That visual alone tells you so much. And the numbers from that study totally back up what we just saw. In individual samples, the caloric content could swing by as much as 29%. But pooling it makes it incredibly consistent. Now for the million-dollar question, what about bacteria? Well, the study found no statistically significant difference in bacterial growth, and that is a huge takeaway. You get that nutritional consistency without a major increase in risk.
So, with that science in mind, how does this all translate into the official guidelines that professionals use for storing and handling human milk?
All right, these are the foundational storage guidelines from the Academy of Breastfeeding Medicine. This is protocol number 8. And as you can see, the pitcher method, which runs on that 24-hour cycle, fits beautifully within that optimal 4-day window for refrigeration. Now, this is a really interesting point. You might have heard the advice to never mix warm and cold milk, and that actually came from the 2017 ABM protocol. The thinking was you wanted to avoid rewarming the chilled milk. But as we just saw from the evidence, research hasn't actually shown this to be a significant safety risk. It's a perfect example of how our clinical advice changes and evolves as we get new data. Okay, but let's be real. No method is perfect for everyone. So to give you the full picture, whether you're counseling a client or prepping for your exam, we have to weigh the pros against the cons. The pros are pretty obvious, right? It saves time, space, and a ton of dishwashing. You get that great nutritional consistency. It's even more eco-friendly. But on the flip side, you have that all your eggs in one basket problem. A single spill or contamination could mean losing a whole day's worth of milk, which is just heartbreaking. And there's also the more nuanced thought that you're blending hormones from different times of day. All right, time to put your knowledge to the test. We've got 5 IBCLC exam-style questions coming right up to see what stuck with you. Ready? First question. According to that 2013 Stellwagen study, what is the primary nutritional benefit of the 24-hour pooling method when you're feeding premature infants? Is it A, it increases the overall protein content? B, it reduces the variability in calories and nutrients? C, it sterilizes the milk? Or D, it increases fat-soluble vitamins? And the correct answer is B. The main benefit, especially for preemies who need really precise nutrition, is consistency. It's not about increasing the average amount of anything, it's about reducing those wild swings between feedings. Okay, question 2. This is a common one you'll get from parents. They're using the pitcher method, but are worried about bacteria from mixing warm and cold milk. Based on the evidence, what's the best way to counsel them? A, tell them to throw it all out. B, explain that research hasn't found a significant increase in bacteria. C, tell them to pasteurize it at home. Or D, suggest adding a probiotic. The answer is B. You can confidently and calmly reassure them by pointing to the evidence. The research just doesn't show a significant risk. The other options are just not evidence-based recommendations for this particular concern. Question 3 is a bit more straightforward, a real know your guidelines question. According to the Academy of Breastfeeding Medicine, what is the optimal maximum storage time for fresh milk in a standard fridge? Is it A, 24 hours, B, 48 hours, C, 4 days, or D, 8 days?
The correct answer here is C, 4 days. This is one of those key numbers you just want to have locked in your brain. It's the whole time frame that makes the pitcher method work so well within the guidelines. All right, question 4. This one gets into one of those nuanced cons we talked about. Blending milk from different times of day could affect the concentration of which time-sensitive hormone that's usually higher at night. A, cortisol, B, melatonin, C, prolactin, or D, oxytocin. And the answer is B, melatonin. You can think of it like this. Morning milk is kind of like a cup of coffee, higher in the get-up-and-go hormone, cortisol. Nighttime milk is more like a cup of chamomile tea, rich in sleep-promoting melatonin. For parents who are worried about this, a simple modification is to use 2 pitchers, one for day and one for night. And our final question, number 5, focuses on those practical, real-world risks. What's the biggest logistical risk of the pitcher method when you compare it to storing milk in individual bottles or bags? A, more freezer burn. B, you can't track your volume. C, losing a whole day's supply in one accident, or D, losing fat content? The answer is C. This is that all your eggs in one basket risk we talked about. It's the ultimate trade-off, right? You get all this amazing convenience, but it comes with the risk of one single devastating loss. It's a really important thing to discuss with families who are thinking about trying it.
You know what this all really shows is that our guidelines aren't set in stone. They're constantly evolving as the evidence evolves. What we recommend is a direct reflection of what we can prove. And it leaves you with a really cool question to think about. As we learn even more about this amazing living fluid, what will we discover next that changes our best practices all over again?